Okay, for part two, our task, as mentioned earlier, is figuring out how to turn light into an action potential. And I want to go back to those three cells that we had drawn in the previous image and see if we can't better understand how they work. As my disclaimer, there are more cells that we're not talking about. This is actually a complicated process. This is a very introductory level of what sight is, but it will give you a sense of what's going on, and that I think for now is going to be good enough. These are our three primary players. We have up on top the photoreceptor cell. And these are your rods and cones. I've drawn it as a rod. They work for the most part the same way. Here in the middle, we've got a bipolar cell. And this one in particular is a bipolar on cell. That's the only system that we're going to be talking about. And then on the bottom, our ganglion cell. What we're really interested in understanding right now is what's going on with this photoreceptor cell. So it has within it what are called membrane discs. And these are continuously being used up and then recycled, and new ones are being made. So they transition, they're made down here at the bottom at the caudal end, and they work this way. So these are new and these are old membrane discs. The disc is shed and it's recycled. We talked about the recycling in the previous movie. That's the job of the retinal pigmented epithelium associated with the choroid. And this full stack is replaced about every 10 days. If we zoom in to this disc, this is what we're going to see. We're going to see a 7-pass transmembrane receptor that we should identify immediately as a G protein coupled receptor on the membrane of this membrane disc. Now, the challenge that we talked about before is figuring out how to get this thing to fire. In all the other cases that we talked about, there was a ligand that bound to the receptor, inducing the conformational change that would lead to its activation. But here, we don't have a ligand. All we have is light. And we need to understand how to do that. And this is accomplished by a little molecule in here. We'll draw it like this. This is called 11-cis retinal. And that cis is important. If you remember from one of our earlier units, we talked about cis bonds and how cis bonds bent the chain. Cis bonds made a chain kinky. And we see that kink right here, right? The fact that it's bent, it's because it's a cis molecule. But what happens is when light hits that, in comes this light, and the light, being energy, has sufficient energy to break that double bond. And when it breaks, it will automatically reform, but during that process, it actually reforms as all trans retinol. And this is important to recognize because it's the change in shape of this molecule, 11-cis-retinal to all-trans retinol, that provides the necessary shifting, the conformational shape change of the G protein coupled receptor. So as this happens, this is how we activate the system. The rest should be simple enough to follow. We have an alpha subunit that was bound to GDP, but following activation of this G protein coupled receptor, we get out the scissors, we cut that off and replace it with a GTP. Beta gamma had been tethering it, right? Here's beta gamma. Hopefully this is all not new to you. That's going to liberate alpha. Now we have alpha bound specifically to a GTP. That's what the receptor did. And in this form, alpha can go to and bind, this time, an enzyme called cyclic GMP phosphodiesterase. Okay, to understand how this allows us to see something, we have to try and understand what's going on in the cell in the absence of light. So let's consider what's going on in the dark. Okay? In the dark, here's the outside of the cell, and we have a channel that's actually being held open by cyclic GMP. It's the same as cyclic AMP, only it's guanine instead of adenosine. It's the same thing. But this is a cation channel. And so remember, this is in the dark. Cations continue to come into the cell. And as cations come in, this leads to a continual depolarization and release of neurotransmitter. So if we come back over here and we look at our neuron, this neurotransmitter being released is glutamate. And in this case, it's delivering an IPSP, an inhibitory postsynaptic potential. So again, in the dark, cyclic GMP, which is being constitutively made by the cell, maintains cation channels open. These cation channels retain the photoreceptor in a depolarized state, leading to the continual release of glutamate. And this glutamate causes an IPSP onto the bipolar cell so that the bipolar cell does not fire. Now, what does light do? Well, if we go back up to what we see up here, the light came in, turned 11-cis-retinal to all-trans retinol, which activated our G protein coupled receptor. This then activated the alpha subunit, which went and bound to cyclic GMP phosphodiesterase. The job of GMP cyclic GMP phosphodiesterase is to take all of my cyclic GMP and convert it into regular GMP, no longer cyclic. And as we do that, cyclic GMP levels drop, and the door then becomes shut. As the door shuts and cations are no longer allowed into the cell stops releasing neurotransmitter. Which means that this signal is eliminated in response to light. Now, this bipolar cell is an interesting cell as well because the bipolar cell doesn't act on action potentials like we've talked about before. The bipolar cell is all about graded potentials. This is very important because action potentials, as you know, are binary. It's on or it's off. And graded potentials allow a more measured response. Think about a light switch. I take the switch and I flip it on or I flip it off. That's action potentials. Think about now a dimmer switch where I have lots of ranges of intensity based on the input coming in. That's exactly how these bipolar cells work. And the likely scenario is very similar to what we saw in the photoreceptor cell, where we have a phosphodiesterase. So phosphodiesterase, what does it do? It takes cyclic AMP and it converts it to regular AMP. And over here we had cyclic AMP that was bound to a channel that was letting in cations. And of course the AMP is going to come around and block that. So in this sense, depending on the amount of cyclic AMP, we can increase or decrease the amount of cations. This is what gives us a graded potential rather than the all or nothing action potential associated with voltage gates. This allows the bipolar cell to release a little bit or release a lot depending on how much inhibition it's receiving, a lot of neurotransmitter, and this happens to also be glutamate, onto the ganglion cell. Now, this is key to understand. The ganglion cell is your typical average neuron. So if the ganglion cell, if we get an action potential, that is going to the brain, and that means I'm registering sight. I'm registering something. But let's walk through the process again of how to get there. In the dark, cations are coming into the photoreceptor cell, causing a continuous depolarization state, and that's releasing glutamate onto the bipolar cell, causing an IPSP to keep the bipolar cell from firing. But as we begin to see light comes in, and on these membrane discs, there's light coming in. It's breaking that double bond here on 11-cis retinal and converting it to all trans retinol. And this conversion, because this little molecule is embedded inside this G protein coupled receptor, this conversion forces this receptor to change shape, and that's what activates it. When it's active, it does what G protein coupled receptors do. It cuts off the GDP, replaces it with a GTP that liberates alpha. Alpha comes over and binds cyclic GMP phosphodiesterase. Once activated, GMP phosphodiesterase takes all the cyclic GMP that have been constitutively being produced inside the cell, it converts it to regular GMP. That essentially leads to this door being closed because there's no more cyclic GMP to keep it open. When that door closes, cations can no longer go into the cell, and that forces the cell into a repolarization phase. When that happens, this IPSP is eliminated. And as I eliminate the IPSP going to the bipolar cell, which is based on graded potentials, has the ability to fire by itself. So I can now deactivate the phosphodiesterase, leading to an increased amount of cyclic AMP, which opens up cation channels, leading to the release of glutamate, which then causes an action potential on the ganglion cell, and that takes that to the brain. Remember, all input goes through the thalamus if we're talking about sensory, except for smell. And the sight is no different, but it's gonna follow the optic nerve through the optic chiasma and then right on to the thalamus, where it's going to be parted out to the various places where this sensory input is going to go.